Hey, hi everyone. Welcome to Everything Turtle Part 2. We're going to be starting today with a student talk from Brooke, telling us about the effects of methodological and temporal factors on efficiency of sampling freshwater turtle communities. And just a reminder for the speakers, I'll give you a warning when you have 3 minutes left, and when you have 1 minute left, I will stand up and start walking ominously towards you, which is your key to please wrap it up quickly so we have time for questions. Okay, I'll also hand over to our first speaker.
Hi, everybody. As Hannah mentioned, my name is Brooke, and I did my master's research at Laurentian University with Dr. Jacqueline Lycodon, and it was in partnership with Magnetawan First Nation. Where's Magnetawan First Nation, you may ask? So, Magnetawan First Nation is in Ontario. They're placed in between Sudbury and Parry Sound. It's kind of northern Ontario, depends on who you ask. And it's a part of the territory described in the Robinson-Huron Treaty of 1850. And it's also a part of the UNESCO biosphere, so this highlights its global ecological significance. And I want to take a second to acknowledge that this land, their word for land there is Aki, belongs to these people, and they're the original stewards of it. And I was lucky enough to work alongside them to develop this project. So for a year and a half prior to starting my master's research at Magnetawan First Nation, I actually worked on staff with them at their Lands and Resources Department. And this gave me the opportunity to learn about what they wanted, what they did, what they already knew, and then kind of work alongside them to co-create this research project. And together we identified what was really important to them, what wasn't super important to them, and then throughout the whole project, we were able to have continual consultation because of that relationship, always checking in if anything changed. They were always checking in if any of their goals changed. And we also built reciprocity into the project by thinking about ways that I could help also with their general programming on top of the work that I was doing. So it was a pretty cool way to start a project, and I'm glad that I got to do it that way. And the research priority that we identified together was to examine the impacts of development on turtle communities. So here you see a highway cutting through beautiful habitats. And unfortunately, it's going to be twinned in the coming future, probably within the next 5 years, on the left side of this picture. So all of that habitat throughout their traditional territories is going to be destroyed for a highway. And they really want to know what's there before it gets destroyed. And we all know turtles aren't doing too hot. So all 8 of our turtles in Ontario are species at risk. And we need to inform their conservation management strategies by learning more about effective population monitoring. So live trapping is a common method that a lot of us have used. Big hoop nets are the most common method. And turtles have low detection probabilities, so this just means they're pretty hard to find. We all know that. It's kind of annoying. And there's a knowledge gap in which way is the most efficient to find them. So we all kind of have our own thoughts and ways that we think are the best way, but when you read into the literature, there's not a lot of papers telling you, like, this is what you should do to get them. And we thought that that could be a pretty cool way to kind of look into that so that when projects are going to happen and you need to locate the populations, that you can. And as we know, well-planned monitoring can reduce the amount of time and the cost of your research. So some of the biases that we know about turtle monitoring is that trapping can have several different biases. So it's known to be biased towards males, it's known to be biased towards adults, and it's known to be biased towards certain species, so usually larger bodied species that can't get out between holes. As well as time of month can affect the detectability of turtles. So if you look later in the year, it's pretty hard to find them if they're in estivation. And the knowledge of the factors that affect sampling design can help us build our programs. So with knowing all of this, and as well as communicating with Magnetawan First Nation, we developed 2 project objectives. The first was just to develop an understanding of turtles in the proposed highway expansion area. There isn't a lot of research done there already, so they didn't know where the populations were and how they can protect them. So that was our first goal, pretty much the most important. And the second was to assess how turtle population monitoring is affected by sampling design. So we were focusing in on both trap type and time of year. To select the areas that we were going to sample, we used long-term data that Magnetawan First Nation has already collected. So they have 10 years of road surveys on the current highway. This is a heat map using those 10 years of data. And we sampled in the adjacent wetlands. So right adjacent to the already hot spots where turtles have been seen on the roads, we were sampling those populations in hopes to locate where they're coming from. And then within that planned habitat expansion, like I mentioned earlier. So that's where we were sampling. And in addition, for my second objective, where we wanted to look at turtle monitoring and what affects it, we used 2 data sets from my lab mates who are also Lycodon labbers and also working in the Georgian Bay area. So Jenna Kentel and Sabrina Lansbury graciously shared their data with me. So we pooled all of our data sets to do that second goal. We actually got 7,682 trap surveys, so a pretty rigorous data set. And we used 3 different kinds of traps to kind of look at how trap type affects sampling design. So large hoop nets, you've probably all heard of these. They're just pretty big fyke nets. And then we also used a small hoop net, which was traditionally used for trapping minnows, so about this big. And then a box trap, which was designed for small body turtles in South Carolina. And we used that here probably for the first time in Ontario. Pretty cool. And overall, we found 772 turtles across the 3 data sets, but specifically at Magnanwan within the area that's going to be developed, 165 of those. Not super great for those turtles, but it's good that they've been found now so that hopefully they can be protected. And this was our spread. So some blandied turtles, which are endangered in Canada, snapping turtles, and painted turtles, which are both special concern. And then to model capture efficiency within these data sets, we use generalized Lexminier models to look at the relationship between capture efficiency, month, and trap type. And capture efficiency was defined as captures per trap check. So each time you pulled up a trap, what was the likelihood that turtles would be in there? And we found that early spring had the highest catch efficiency. So you probably all could have predicted this, but it's cool that we could actually prove it. So what this graph is showing you, the blue is the confidence intervals, and the red is showing you the difference between the groups. So if there's no overlap within the red, it means that they're significantly different. So here you can see that May and June were the best months to trap. We predict that April probably could be good, but we only had 1 data set that trapped in April, so pretty wide confidence intervals there. And August and July were pretty sucky for trapping. Water levels are pretty low then, and I'm sure if you've trapped in August or July, you know it sucks. And why spring may be best for trapping? So activity is usually increased in the spring. This is for habitat and mating behaviors, more chances to encounter a trap if you're moving more. And then also limited food sources in the spring could be bringing them into traps because they were all baited with sardines. And then we also found that trap type influenced the species that were captured. So this is the same kind of graph that I just explained. So the red is showing the differences between groups. Surprisingly, we found that box traps, which was the new design that we built, were significantly better than the rest of the traps for catching painted turtles, as well as juvenile turtles and any hatchlings. So they were the only traps that were successfully able to capture turtles under 3, which was pretty cool. The holes are pretty small in them, and it's metal, so there's no movement. The turtles can't squeeze in between them. And then for both snapping turtles and blanding turtles, we found that overall the large hoop nets, which is what's been used traditionally, were best. But we think that using the box traps are still really valuable because there's taxonomic biases and potential age class biases to be looked at. So if you're just using the large hoop nets, you might not be trapping a lot of the smaller bodied turtles by species and by age class. So your juveniles and your hatchlings could be completely excluded from your sampling design, which could have serious results if you're looking at a whole population. And then why this might be influencing, as I just mentioned, the entrance holes and the mesh sizes are what's dictating what can get in and out of your traps. So turtles are pretty smart, and I think that the hard-bodied traps are a pretty cool design that maybe prevents them from getting out. So the implications of this study are that trapping in the early spring is probably most efficient. We all knew this, but it's kind of cool to look at it with data. And then trap type will have large impacts on what species you catch and what size of turtles you catch. So we recommend that most people, if you're trying to sample a whole population, use varied trap designs to allow you to capture the whole breadth of the population. And then also we just want to highlight that collaborative research yields results that are important to the community. So the first goal of this research and the most important goal was to provide Magnanwan First Nation with the data that they wanted, which is the 165 turtles that we found within that development area. And this will help them inform decision-making and land management as the developments come in the future. And something that's really important and we have to talk about is data sharing and research agreements, because this data doesn't belong to me, it doesn't belong to our lab, it belongs to the First Nation who wanted this research to be done. So we created a research agreement based off of OCAP principle, which highlights ownership, control, access, and possession of data. And some of the strategies that we incorporated into our research agreement, which should be tailored to your own project. So if you are interested in it, I recommend reading Shark 2004. But that the project is community initiated was a real high priority for us. We want the community to be interested in what we're doing, and we want it to be valuable to them. Relevance and benefits for both parties. So not only was I able to write about this and we were able to learn more about turtle population management, but also they know a bit more about how they want to protect their lands. Cultivating long-term and respectful relationships. So Dr. Lycodon, has been in relationship with Macnabon First Nation for over 12 years. And I didn't just come in here out of nowhere also. I created a relationship with them over 2 years. And kind of something James talked about last year, which I thought about a lot, was the parachute approach, just dropping in and going out. It's not super beneficial, and I think that your project can have a lot bigger of an impact if you really have those relationships. It helps you know what they need and what they want. And then the research outcomes will always be reviewed by Macnabon First Nation. So they reviewed this, they'll review anything that I publish and make sure that it's okay with them. So I recommend looking into this if you're doing partnership projects. And then reciprocity, something I mentioned at the start of the project, at the start of the presentation. These are a few different ways that we incorporated it into our project, but there is a million more ways you could do it. So for knowledge co-production, we made the decisions together throughout the whole project, and any literature that's published will be co-authored, including this presentation. And then maintaining data sovereignty and rights, so our research agreement and the data ownership. As I mentioned, the data is owned by them, and ultimately they get to decide what is done with it. And then also sharing research in accessible ways. This one is really important for the community because they don't want the research that I do to just disappear and the community to never hear about it. So we did some science sharing series where community members could come and listen about what we did and why they might care about it, which was really fun. And kids were really into it, so I love that. And then newsletters and research summaries are shared pretty much continuously throughout the project so that the community could read about what was changing, what wasn't changing. And also fostering science in future generations, something that I'm pretty passionate about. Throughout my project, we had 4 or 5 youth members that were pretty much coming out with me weekly. They really liked sampling turtle populations because it got them out of the office. And they liked tramping around in the bogs, and they knew a lot more than I did about where turtles would be sometimes, since they've been tramping around in the bogs for a lot longer than I have been. So that was a really fun part of our project, and I'm grateful for them. And that's everything that I have to talk to you about today. Thank you, Miigwech.
Great talk, Brooke. As always. I had a question about the box traps.
Yeah.
Because they seem pretty groovy.
Mm-hmm.
How do they work, like, logistically? Like, I can collapse a big hoop trap, pretty small, load a bunch into a canoe, and take off.
Mm-hmm.
Are they collapsible? Like, what's the field logistics of using them?
Terrible.
Yes, thank you.
Yeah. So I built these based off of the paper that designed them, which was a really fun process, but they end up about this big, and it's hard metal, pretty heavy, and I carried them out to the wetlands, and luckily the 8 wetlands that I was trapping in were the same wetlands the whole year, but if you were doing a rotational schedule, it could be pretty annoying. So that's something to consider too. Yeah, they're pretty big and they're pretty heavy. The bonus is, though, all the other traps, I was repairing them basically bi-weekly, like animals and different things eating through them. These, over 3 years, 0 repairs. Bears would try and get into them, and they would just make little dents. So that's a bonus. Yeah. Yeah, thank you.
Great talk, Brooke.
Thank you.
I was wondering if you're, during your literature search or discussions with Jenna and Sabrina, if you think that using a combination of different types of bait would have any sort of effect on how many turtles you're getting, even throughout the active season, if you're changing the bait throughout the different months, would that have any sort of effect?
It's a really good question. And post all of our projects, I think we've all concluded that changing baits could be something to look into. The problem is if you change baits, it might affect your like study design, so it probably would have to be its own project. There's a really cool paper about using buffalo chicken to trap turtles, and it's super efficient, better than the rest of the things that they tried out, so I recommend reading that.
Great, now I'm gonna get stuck in.
Yeah. But we all use sardines, and Sabrina used some dog food too. But I think that bait choice could be pretty significant. Buffalo chicken. Yeah.
Okay, so thank you so much, Brooke. Next up, we have another student speaker, Steph, who will be telling us about population demography and spatial ecology of spotted turtles.
Okay, hi everyone. My name is Stephane Thibeault. I'm a master's student in Jackie Lycodon's lab as well, at Laurentian University, Sudbury, Ontario. Didn't mean to do that. Never happened. And I'm also working in partnership with Scales Nature Park, founding the STAR project to study the population demography and spatial ecology of a central Ontario population of spotted turtles. Let's just get right into it. Basically, studies of population demography and spatial ecology give us good ideas of things like population structure and movement dynamics respectively, right? So when looking at population structure, you're looking at sex ratios, age ratios, growth and decline trends and abundance, right? Moving to movement dynamics, you can look at distribution, migration, as well as home range. But why do we do these studies? Well, they give us really good ideas of what the natural and life history traits are of specific species, and then these give us ideas of how to go forward with conservation if they're at risk, right? And then sometimes you require site-specific conservation for con-specific populations that vary in the environmental pressures that they're subjected to. So this kind of brings me to our study animal, the spotted turtle. So it's a small species of freshwater turtle endemic to eastern North America, and they are endangered globally, federally, and provincially in Ontario. And literature shows that actually there is variation in traits across its range, but also across habitats that aren't necessarily in different latitudes, right? And we know a lot about the coastal populations of Ontario, but we don't know a lot about the inland populations, right? So we need to find out more about these to help inform those site-specific conservation decisions. So I worked in partnership with the Saving Turtles at Risk Today project, the START project, founded by Scales Nature Park, which is an Ontario turtle research and conservation project, and they actually discovered 17 undocumented inland populations of spotted turtles in central Ontario. And on one of these, it's a 50-hectare sphagnum bog, they've done about 9 years of surveys, which kind of brings me to the aim of my study is to describe the population demography and spatial ecology of this specific inland population. So the first objective, looking at population demography, we're looking at demographics like sex and age ratios, adult size dimorphism, as well as abundance and density. And the methods I used was basically, we had that 9 years of mark recap data from the STAR project that we used, and then I also created a closed population model to look at abundance. The second objective, looking at spatial ecology, we're looking at attributes like home range, daily distances moved, habitat use, and then different behaviors, as well as the water depth selection. And then the methods for that, of course, was a 2-year radiotelemetry study where I followed 20 turtles, 10 males, 10 females, and then tracked them about 2 to 3 times a week. And the third final objective was to basically compare the demography and spatial ecology attributes to those of Ontario's coastal populations that we already know so well. So let's just kind of go into the results and also discuss some of these results at the same time. So looking at the sex ratio, abundance, and density, just specifically looking at the START data that we have from 2014 to 2024, they marked a total of 127 individuals, 121 of which were adults. Now, the sex ratio for that was about 1.5 male to 1 female, so that's 60% male, 40% female in total. And then the closed population model that I created, that only included 4 years of that START data because that's where we had the most consistent survey effort, so it was suitable for the model, and it included 65 of the 121 adults that we've marked over that 10-year process. And the population size it calculated was about 92 plus or minus 10 individuals. And then we got a density of about 1.84 individuals per hectare. So basically, what does that kind of mean? So we have a male biased sex ratio here. And from what we know of this species, the literature usually reports either an equal sex ratio or female biased. There is one example of a male biased population in southeastern Ontario. However, road mortality of nesting females plays a big role in why we have that male biased sex ratio. And for our population, we don't have a lot of anthropogenic activity, like nearly none. So it's very unlikely that that is playing a role in that sex ratio that we found. Now, one possibility is that males were more readily caught. For the STAR project, we survey usually in the springtime, and I will get to another result that might explain this a little bit more, okay? So looking at sexual size dimorphism, this is a lot of numbers, but basically what I want you to focus on are these two carapace length measurements. So it showed that males had significantly larger carapace lengths than females in this population. And then females had significantly larger carapace height, as well as P-max measurement, which is one of the plastron length measurements. But specifically focusing on those carapace length measurements. So differences in carapace length are pretty uncommon for this species. We do have one example from an island population where the males had the bigger carapace length. However, there was a pretty low sample size in that study, so they couldn't really make any sound conclusions. So males had about 1% larger carapace length than females in our population, which interestingly actually matches the Lycodon et al. 2004 paper that looked at clinal trends in body size for this species across its whole range. And it showed that males overall had about a 1% greater body size than females. So that's pretty cool. However, that small difference is probably not significant biologically, while it is statistically, right? Now, moving on to sort of spatial ecology here and looking at the home range area. So from what you can see here is that males had way bigger home ranges than females. So it was significantly larger. It was about 77% larger than females. Now, what we know about coastal populations, our home ranges vary from about 3.2 to 7.9 hectares. For the inland populations that have been studied, very few. It's about 1.6 to 3.7, and ours fell in between that range at about 3.1 hectares as a mean value. So males had a larger home range than females, which for this species has actually never been reported in the literature. So that's a pretty interesting result. Usually what we see is about an equal home range, or it tends to be larger for females. So looking at the movement patterns now across these seasonal periods, we have emergence here, which is April to May, and then nesting June, estivation July and August. We can see that after an ANOVA was done, so sex and seasonal period both had significant effects on daily distances moved. Their interaction was not significant, and then the post hoc test showed that significantly greater distances were traveled during emergence earlier in the year. And then overall, looking just at the sexes, you can see that males, again, traveled significantly greater distances per day than females. So someone must have put that in there. I don't know what that is. And so looking at the movement patterns, males travel greater mean daily distances, right? Even during the nesting season, which is pretty uncommon. Usually females will match or exceed male movements during this period. So is it possible that we underestimated female movements and hence home ranges? So this is possible. There was decreased tracking in the month of June because I got COVID. And so we were doing regularly 2 to 3 times a week, and then it went to about 1 to 2 times during the month of June, right? But another possibility, which is purely speculative, is that are females nesting on the bog? So we weren't successful in finding females nesting anywhere at our site because 1, it's pretty dangerous, and 2, we only saw 1 female actually going on land during the month of June, which is pretty surprising. And there's some anecdotal observations of females nesting on bogs in other populations as well. So this could be a possibility which would lead to that decreased home range and movements as well. Then looking at habitat use, basically all that gray you're seeing there is all use of bog habitat, and they hardly use any other habitat. So you see some bog supremacy here. And then doing a pairwise Fisher's exact test to show that there was significantly greater use of bog during estivation relative to emergence. So estivation being July and August, emergence being April, May, right? And then looking at some more habitat use, specifically water depth use, we saw that turtles are found in significantly deeper water during estivation relative to emergence, and then pre-hibernation, which is sort of after, this would be September to about October. So very little terrestrial activity going on here. They're mostly using the bog throughout the year, it seems. And bog estivation was something that was seen a lot in this population, and this has actually been seen inland before in another inland population. So it's possible that this is a common thing in inland populations. Hard to know right now because there's only about 3 studies, including mine, that have really come out about this. But for water depth, we saw an increase in the amount of water that they were in with every seasonal period. So we saw it going up as it got to estivation. And what we usually see in coastal populations is the deepest water measurements being in emergence, and then it decreases during estivation because in coastal populations, they tend to go onto land to estivate. And then it increases again in pre-hibernation, but we saw the complete opposite of that. So that's pretty interesting as well. So it's possible that maybe the bog has some more preferable thermal qualities than terrestrial estivation would, but it's really hard to know because the proximate factors that influence estivation and influence those behaviors are very poorly understood. So there's a lot more research that needs to be done on that. So in conclusion, we successfully described the population demography and spatial ecology of this inland population. And in it, we saw that males exhibited somewhat novel trends with relation to sex ratio, body size, home range, and movements, right? And then we also saw those reversed water depth trends as well, which is pretty interesting. And then for the conservation implications, we see that bogs are very important habitats year-round for these populations. So that's something that we kind of need to take note of, right? And for future research, I suggest someone to research the nesting ecology of females in inland populations to better figure out what exactly they're doing during this period, right? And then, of course, trying to figure out the proximate factors that influence estivation and those behaviors is also very important because we really have no clue what's going on with any of that. Yeah, I'd like to give a special thanks to, of course, my supervisor, Dr. Jackie Litzkis, my field tech, Doug Warner, as well as Jeff Hathaway and Kelsey Moxley back at Scales Nature Park. Thank you very much.
Yeah, I had a question about the sex ratio.
Yeah.
So I also sometimes do work on these bog mats catching painted turtles, and I've noticed that under the like under them, just a few centimeters, it can be really, really cold.
Mm-hmm.
Do you think the bias in sex ratio might be because then if these females are nesting there, it would probably be cooler temperatures, which would likely make them more male?
TSD, right? Yeah. So I was watching your presentation earlier and I was thinking about that a little bit because we're kind of unsure why that sex ratio is there because it's not anthropogenically caused. We know that. So it is possible it could be TSD. But first we sort of need to figure out whether those females are actually nesting on the bog, right? That's one of the future research directions we should see. But it could definitely be that. I'm not sure, but it's a great question. Yeah.
We have time for another question. Nothing online. Okay, go ahead, James.
All right. Great talk, Steph.
Thank you.
I had a question about, and without spilling obvious secret beans, we're talking about spines, right? The structure of the habitat. So like sometimes I've noticed that you get males that go for big wanders when it's more kind of patchy. There's a wetland patch, and then they got to go upland and go for a friggin' hike, and then they get to another one, and they kind of move through a landscape. But if you're seeing kind of shifts, is it more of just a continuous bog? Like, what's the nature or like the structure of your wetland?
Yeah, so basically the structure is basically this continuous bog, basically a 50-hectare bog, and it's encircled by rock outcrop, basically. And there's some wetlands on the other side of some of the rock outcrops, but basically the turtles I was studying, they were mostly hanging out in that big bog, right? Yeah. No problem.
Okay, thank you, Steph. We're going to move on to our next
Thank you.
speaker now. Okay, next up, we're going to have Heather telling us about monitoring the response of turtles to Phragmites management.
You're live.
Hi, everyone. I'm Heather, and I'm a biologist at Natural Resource Solutions. And we're going to talk about Phragmites. So let's just quickly refresh ourselves on why we're so keen on managing this species. It's an aggressive, non-native, invasive grass species. When it establishes, it establishes as a tall, dense monoculture and displaces native wetland species and communities, resulting in an altered wetland habitat structure and reduced plant diversity. And keen to our hearts, reptiles and amphibians have been shown to be negatively impacted by the species. Previous studies have reported significantly fewer observations of reptiles and amphibians in Phragmites-dominated areas compared to areas with little to no Phragmites. Other studies have reported on impacts on turtle nesting habitats, as the tall species will shade out typically sunny open nesting habitats, changing nest site microclimates and potentially influencing turtle hatchling success. Other studies have reported on the avoidance of Phragmites stands by Blanding's turtles, except during nesting migrations, where they've been tracked through the Phragmites to access nest sites. And the decline of Fowler's toad has been linked to the colonization of shallow aquatic breeding habitats by Phragmites. So Phragmites is bad, and we should be managing it. But we need to make sure that the approach that we take to managing Phragmites is effective and doesn't result in adverse impacts to the ecological communities that we're trying to protect. So we set out to answer the question, what is the impact of Phragmites management on turtle relative abundance and habitat use? We developed or designed two main objectives. The first, to evaluate relative abundance and habitat use of turtles prior to and following management through visual encounter surveys. And the second, to evaluate habitat use of turtles prior to and following management through radiotelemetry. Unfortunately, I don't have time to talk about both of these objectives today, so we're just going to focus on the first one. So our study site was in Ontario, along the Lake Erie shoreline and on the Long Point Peninsula. The Long Point Peninsula is a very special place. It's our largest freshwater sandspit marsh complex of the Great Lakes. It was designated as a World Biosphere Reserve, and it provides critical habitat for 25% of our species at risk. The Government of Canada owns and manages 2 national wildlife areas on the Long Point Peninsula, including the Long Point National Wildlife Area and Big Creek National Wildlife Area, both within which we conducted our study. The peninsula was colonized by Phragmites in the mid-1990s, and since then it has increased in area by about 15 to 30% per year. So to meet our first objective, we conducted a before-after control impact study. We selected 4 main sites, 2 of which were selected in 2019 during a pilot study, 1 within the Big Creek National Wildlife Area and 1 within the Long Point National Wildlife Area. Our other 2 sites were selected in 2021, again, 1 within the Long Point National Wildlife Area and 1 within the Big Creek National Wildlife Area. Within each of those 4 main sites, we selected 5 treatment habitat patches and 5 control habitat patches. with the exception of the new, with the exception of the pilot Long Point National Wildlife Area, where we had 7 treatment habitat patches and 7 control habitat patches. Our treatment patches consisted of areas dominated by Phragmites that were managed during our study through herbicide application and mechanical management. And our control habitat patches also consisted of Phragmites dominated areas, but were not managed during the study. Within each of these habitat patches, we conducted visual encounter surveys following methods that were implemented during the pilot study. Our survey period was between April 1st and June 30th on each of our monitoring years. And within each habitat patch, our search effort was 3 person-hours per hectare. Each of our habitat patches were surveyed a minimum of 3 times each monitoring year, with the exception, again, of the pilot Long Point National Wildlife Area, where each of the habitat patches were only searched twice per year. During each of our surveys, we documented the number of turtles and species captured, and we collected information on 5 habitat variables, such as average water depth and average vegetation density. So to jump into our results, this graph is showing the mean number of turtles captured per habitat patch type per monitoring year at our pilot sites. The vertical dashed line represents when our treatment habitat patches were managed for Phragmites. The error bars represent standard error. So there's a little bit more information on here than we need to focus on for this presentation. I want you to focus on just the treatment and control habitat patch data. In our pilot Big Creek National Wildlife Area, we found that turtle abundance was significantly reduced post-management. And within our pilot Long Point National Wildlife Area study site, we didn't find any significant difference between our treatment and control patches within any given year. You'll note that we did have a significant reduction post-management, but that reduction was within both of our control and treatment habitat patches. At our new sites, and specifically within our new Big Creek National Wildlife Area study site, again, we didn't see any significant difference between our treatment and control habitat patches within any given year. But in our new Long Point National Wildlife Area study site, we found that turtle abundance increased significantly more in our treatment habitat patches versus our control habitat patches post-management. Okay, so we have some contradictory results. And I just want to dig into a couple of them with you. Specifically, the pilot Big Creek National Wildlife Area, where our results indicate that Phragmites management has a negative impact on turtle abundance. But perhaps this might have something to do with turtle habitat preferences and the presence of and structure of vegetation post-management. As we know, most of our turtle species in Ontario prefer shallow aquatic habitats with abundant submerged and/or emergent vegetation. A number of our treatment habitat patches in this study site, we consistently documented them as open aquatic habitats lacking any sort of vegetation post-management. So it's possible that high water levels prior to 2022 in Lake Erie likely prevented the reestablishment of Phragmites post-management, but probably also impeded germination and growth of native vegetation in these treatment patches as well, reducing habitat suitability for turtles and resulting in the potential avoidance of these treatment habitat patches by turtles. At our new Long Point National Wildlife Area, where our study results indicate that Phragmites management has had a positive impact on turtle abundance, this was likely driven by the fact that we didn't have any turtle observations in our treatment habitat patches prior to management. As you can imagine, based on these images, it was a bit of a challenge to search for turtles in dense Phragmites stands prior to management. But it's also interesting to note at this study site, we observed a higher rate of vegetation establishment compared to our other study sites. For example, the image on the left is from our new Bay Creek National Wildlife Area study site, 2 years post-management, and we're still seeing a dense mat of dead Phragmites with little to no vegetation establishment. The two images on the right are from our new Long Point National Wildlife Area study site, and we're clearly seeing some vegetation establishment post-management. It was also interesting that the new Long Point National Wildlife Area was the only study site that we observed stable water levels at, which may have contributed to the establishment of vegetation at this site as well. So in conclusion, our study results don't really tell us much about how Phragmites management impacts turtle abundance. It doesn't seem that bad, but it also doesn't seem that great. So I think I would like us to consider options for improving how we're managing Phragmites, and specifically at this study site, we might want to consider follow-up Phragmites management. We were noting that at a lot of our habitat patches, we were seeing dense mats of dead Phragmites post-management. And maybe something that we could consider is removing some of that Phragmites litter to help support the vegetation establishment post-management and the recovery of these habitats quicker post-management as well. In the end, the effects of management on reptile communities may not be apparent without long-term monitoring. And I know we say that for everything, but I think in this case, it is extremely relevant. Okay, with that, I would just like to thank absolutely everyone that's been involved with this project. There were quite a few of us conducting visual encounter surveys over the years, so thanks to everyone that also came today. Thank you.
Okay, we have time for a few questions. Anyone? Yeah, Jackie?
Thanks, Heather. You didn't have time to talk about it, but can you mention some general trends from the rate of telemetry part?
Sure. So with regards to the rate of telemetry, unfortunately, we didn't have too many turtles that we tracked from one year to the next. But it was interesting that a few of them did show their home ranges shifted a little bit away from areas that were managed for Phragmites, which somewhat aligns with some of the results that we're seeing through the visual encounter surveys. Other individuals, their home range actually expanded post-management, and I don't know if that was a result of perhaps habitat suitability was reduced within their home range following management and they needed to explore a little bit further, or because management allowed them to move a little bit further. But yeah, it was interesting to see that, even though it was only a few individuals. Yeah.
Okay, thank you, Heather. We're going to move on to our next two talks now. Okay, the next two talks are going to be lightning 5-minute talks. So there won't be time for questions between them, but I'm sure you can find them afterwards with all of your burning questions. And first, we're going to be hearing a student talk from Tyler, talking about sexual size dimorphism in an endangered turtle.
Hi, I'm Tyler Ambeau, and on behalf of my co-authors, I'd like to present Life in the Third Dimension, Sexual Size Dimorphism in an Endangered Turtle. In many species, body size varies geographically, with a few biological rules that have been proposed to describe these trends. Bergman's and James's rule describes a trend of increasing body size with latitude, both within and between species. The temperature size rule describes a trend in ectotherms of increasing body size with decreasing temperature, but this trend is inconsistent in reptiles, especially in turtles. Within a species, if rate of increase between the sexes, varies between the sexes. Sexual size dimorphism can emerge. Trends in turtle body size have had mixed findings. Generally, within species, body size tends to increase with latitude, but between species, body size tends to decrease with latitude. The trends can vary between species and regions. Now, latitude is just a number that we made up so we can navigate the world. So what's actually causing these associations? Those associations can be weak. Temperature is currently the best predictor of turtle body size. One potential explanation for this is that larger body sizes are more efficient at retaining heat, also called thermal inertia. Differences between the sexes in turtles can arise from selection for increased reproductive success, fecundity, and niche partitioning. In this study, we work with spotted turtles. There's evidence of larger body size in more northern populations of spotted turtles, but latitude was not found to be an accurate predictor. And in one of Jackie's papers, the trends were also found to be nonlinear, so I'm digging more into that. So I chose to explore what factor might explain the observed trends by testing the hypothesis that if increased thermal inertia increases fitness in colder environments, then selection should favor larger turtles in colder environments. I'll also be examining differences between males and females straight carapace length and body and shell height to determine if sexual size dimorphism exists in spotted turtles, in which case more work can be done to understand why. There are a few studies that use more than one dimension when examining sexual size dimorphism in turtles, and I believe this could be a mistake as it could be creating false negatives. After all, why would we not treat a three-dimensional animal as such? Turtles were caught by hand during weighting surveys. This analysis includes 1,140 individuals from across 12 populations from southern to central Ontario. Most recent measurement was used for analysis for each individual. Shell height data was analyzed for 11 populations, and shell height was also scaled by straight carapace length to account for exceptionally large individuals. Climate data came from Climate Atlas of Canada. And it's important to note that male shell height measurements did not include plastial concavity as seen in this diagram. So looking at this graph with straight carapace length on the y-axis and average annual temperature on the x-axis, we can see that average yearly temperature did not predict straight carapace length, nor did it predict shell height. Moving on to sexual size dimorphism. On the y-axis, we have straight carapace length. On the x-axis, we have site codes arranged by average annual temperature, as in the previous graph. We have females in orange and males in purple. When examining straight carapace length, there's no clear evidence for sexual size dimorphism, as significant differences were found in only 5 sites, and the direction of these differences varied. However, looking at this graph, we can see that carapace height, on average, is greater in females than it is males by about 5 millimeters. And I think this result illustrates the importance of considering multiple dimensions when analyzing sexual size dimorphism in turtles, or really any species. So in conclusion, there's evidence for sexual size dimorphism in spotted turtles, especially when looking at carapace height, but it's a bit more ambiguous when looking at straight carapace length. And temperature do not predict the variation in body size between sites, but we really need to look at growth rates to properly test this association. And that's what I'll hopefully be presenting at the next CHS, so stay tuned for that. I'd like to first thank the original and ongoing stewards of Turtle Island, without whom this project wouldn't have been possible. And I want to thank a particular community who asked not to be named because of location sensitivity, but you know who you are, and I greatly appreciate your contribution. And a thank you to all the organizations that donated funding and/or time. And without the countless field staff and volunteers, this project would not have been possible. So thank you to them as well. And we'll just wait a second. Last but not least, a thank you to all my wonderful co-authors. And that's it. Thank you. Oh, yeah, sorry. Right.
Thank you, Tyler. Next, we have Hinrich, who will be telling us about a potential genetic explanation for unusual color morphs.
This is not about turtles. Please stay. So I just wanted to say that most of you probably have heard about snake polymorphism. This is now somewhere completely different. This is in India. And I'm working with my new graduate student, Dikansh Parma, on this topic, and he's been involved in snake rescues. And you see a lot of interesting things when you rescue animals, don't you? And so this is in places in India called Gujarat and Goa, illustrated on the map. And one of the things that you probably all know is that snake polymorphism exists all over the place. There are many different patterns, and I want to introduce just a couple of them. There's, of course, the albinos, and then you'll have the ones that have some pigmentation in them. These are called flavinism. We have leucism, where just the eye is black. And then, of course, you have a huge number of different patterns when it comes to pythons. And most people who know about the pet trade know that these pythons can be all kinds of interesting patterns. And so what Dikanch found when he was working with snake rescue people, they rescue all kinds of interesting snakes. This is a typical wolf snake from one of the areas that he did research in, and you can see it has very nice bands. Now another one turned up and it looked like this. This is also a wolf snake, and you can identify it by the way the head looks. The characteristic color and the pattern is there, but then the color, the pattern around the rest of the body is completely different. So I look at this and I think, why? And so then, of course, more snakes get rescued. This is a normal Daboia russelii, that's Russell's viper, which is one of the most dangerous snakes and one that kills the most people, probably one of the ones that kills the most people in the world. This is normal, and you can see the blotches. And then you have these. And again, the question is, why? And so clearly, there must be a genetic connection. And the problem with that is that we don't really know a lot about this. So part of this discussion is simply to bring this to your attention, and maybe when you see some odd snakes, you might pay attention to them. The genetic connection is that there are some candidate genes. This all has to do with the epithelium and how the skin and the scales are formed in reptiles, and we really don't know that much about it. There are now some candidate genes for how striping is working and how color is working in general. This is in pythons, but it turns out that these genes are actually highly conserved in vertebrates. That's all vertebrates, including you and me. And so this is one of the interesting things that people are now trying to discover more of. So if you see anything that has aberrant coloration, it might be a good idea to take a scale snip to actually allow some of this genetic analysis. Of course, the snake rescue people in India, they didn't think to do this, but hopefully now they will know about this. So how do we actually deal with this? Well, humans are actually pretty good at pattern recognition. And so what we did, Dikanch and I, is we actually took a look at all the different kinds of patterns that had been identified with the genetic component in these pythons. And so one of the things I was trying to figure out is, well, what if we just went through this list and eliminated those kinds of things that didn't look anything like the animals we found? And so then we would basically look at what we need. That's at the bottom right. And so we say, okay, no albinos needed. We don't need any of the ones that have blotches or anything remotely looking like blotches. We can eliminate anything that has blotches and stripes. So what we're left with is just a few genotypes that might work for this. This is conjecture. This is not something we've been able to prove yet, but nonetheless, it's something really interesting. And so if you look at this one here, you can see that the spark genotype, hence the name of the talk, is actually pretty close, pretty identical to the one we found in the viper. And so ultimately, when it comes to these kind of things, you can see that there are a lot of these similarities. These are the snakes that we were interested in. The spark fits the pattern. The striped vipers have a little bit more pigmentation, so maybe there's a different combination. There are certain candidate genes that we've identified. And interestingly, the color of the head seems to be decoupled from all of this because the head still shows the pattern you might expect. So what's the conclusion to all of this? Well, the conclusion is that nature is a sometimes eccentric painter of lines. Thank you.